Good morning dear students and welcome to Genesis. Well, in this class we will finish the chapter pollination and fertilization with the last topic, which is fertilization. So what do we understand by the term fertilization? Fertilization is fusion of the male gamete or the male sex cell and the female sex cell, right? Now, so in case of plants, let us try to find out the structures that produce the male and the female gamete. So in case of plants, the pollen grains, they actually produce male gamete. And so let us try to see the structure of the pollen grain. The pollen grain is basically a unicellular structure, but it has two covering membranes. The inner one is called intine and the outer layer is known as exine. Remember one thing, intine is continuous but exine is discontinuous, right? And inside this there should be a nucleus like it is present in every cell, but instead of one nucleus there are two in case of a pollen grain. So let us label the structure. The outer structure is called exine. The inner membrane is known as intine. Then there is one nucleus which is smaller in size and it is called the tube nucleus. Or this is also known as vegetative nucleus. Why so? Because this does not directly participate in the process of fertilization. And right next to it is the other nucleus which is known as the generative nucleus. This directly participates in the process of fertilization. How? Because this will divide to give rise to two sperm nuclei. So this produces two sperm nuclei. So from one generative nucleus, remember this, there are two sperm nuclei or two male gametes produced. Now let us understand why the outer layer of pollen grain is discontinuous. Because when this pollen grain settles on the stigma of a flower of the same species, if it is of the same species, then this pollen grain gives out a tube-like structure which is known as pollen tube. How is this possible? Because on recognition, the stigma produces certain chemicals which are sweet and nutritious, and the pollen grain consumes that nutritious chemical and then directs the pollen tube. Now, for the development of the pollen tube, this discontinuous exine comes in use. So let us draw a structure where the same pollen grain will start to give out a pollen tube. So when this gives out a pollen tube, the intine extends through the opening between the exine. And what is contained inside the pollen tube? There is number one, the tube nucleus, because its only function is to elongate the pollen tube, and the two sperm nuclei which were produced by the division of the generative nucleus. So this is all we have to understand about pollen grain before we understand fertilization. So the pollen grain is unicellular, covered by two layers, outer one exine, inner intine. There are two nucleus inside. The bigger one is called the generative. It directly participates in the process of fertilization and divides to produce two sperm nuclei. And the other one is called tube nucleus. It does not directly participate in fertilization but helps in the elongation of the pollen tube. Now the female gamete in plants is produced by a structure called ovule which remains inside the ovary. So the ovule produces female gamete, and now we have to study the structure of this. So if you look into the structure of gynoecium, it has stigma, then it has style, and then there is ovary. So this is stigma where the pollen grain will come and settle. Then it is style through which the pollen tube will go, and here is the ovary, and inside the ovary there is ovule. There can be one ovule, there can be many ovules. Now this pollen tube has to basically reach the ovule. Why? Because inside the ovule the female gamete is present. So now let us understand the structure of the ovule in details. Well, the structure of ovule is so that it is lined by two protective coverings. So this is the first covering and here is the second. Similarly, this is the first one and here is the second one, and this is how it makes an envelope around the ovule. These coverings are known as integuments. So this and this, they are integuments. So there is one inner integument and there is one outer integument. The inner integument is called testa and the outer integument is called tegmen. Now within this, the entire mass of nutritive cells are present and they are known as nucellus. Apart from this, you can also see there is an opening here between the integuments. This opening is known as micropyle. Now, it basically serves two functions. One, when this ovule later on after fertilization becomes seed, this micropyle acts like an entry point for water. So, whenever you soak seeds in water, you would see the seed swell next day. Now, if the seeds are completely covered with seed coat, then how can water enter inside the seed? It enters through the micropyle, and these integuments, the inner and the outer integuments, they are the seed coats. Now, like I told, the inner mass of the ovule is called nucellus, and it is made up of nutritive cells. Right inside this, there is another sac present, and this sac is known as embryo sac. And there are many structures inside the embryo sac. Well, there are 3 cells at the top opposite to the micropyle. These cells are known as antipodal cells. And there are 3 cells with single nucleus. Again, there are 3 cells at the lower end near the micropyle. Now here, the central cell is known as the egg cell, which is the female gamete, and this egg cell is flanked by 2 cells on either side, and they are called synergids. So again, there are 3 cells with 1 nucleus each. Right in the middle, there is another cell, and this is called the central cell. But the best part here is this central cell has 2 nuclei, and they are known as polar nuclei. So when you look at the structure of the embryo sac, you can see that there are 6 plus 7 cells. So there are total 7 cells, but there are 3, 6, 7, 8 nuclei. So this is why we say that the embryo sac depicts a 7-celled 8-nucleated structure. Now, with all these structures, which is the most important structure is this one, because this is the female gamete. So, in this diagram, imagine if I draw the embryo sac inside this with antipodal cells, the central cell right in the middle, and the egg and the synergids. Now, you will be able to relate what basically is going to happen now. Inside the pollen tube, there are 3 nuclei. That means there are 2 sperm nuclei and 1 tube nucleus. The tube nucleus is going to degenerate as soon as the tube enters through the micropyle. It knows that my function is over. My function was to bring the tube close to the egg, which I have already performed. So I can degenerate now. Now we are left with 2 sperm nuclei. Where are they going to go? The first sperm nuclei will enter through the micropyle and fuse with the egg. So here will be egg plus sperm nucleus 1, and whatever structure that gets produced is known as the zygote. This zygote will later on divide and redivide and will form the embryo, and the embryo will develop into a baby plant. On the other hand, the next sperm nucleus, which is the sperm nucleus 2, will go and sit with them. So they are going to sit with the polar nuclei and will form a triple N structure, which is N plus N plus N. This is a triploid structure. This process where there is fusion of 2 nuclei along with 1 nuclei, this is called triple fusion. So now if you look at this entire concept of fertilization in plants, it is little different from fertilization in animals. In case of animals, the egg containing 1 nucleus fuses with the sperm that also contain 1 nucleus, and the resulting structure is a zygote. But in case of plants, there are 2 fertilizations happening. The first fertilization, where the sperm nucleus 1 goes and fuses with the egg and produces zygote, just like in our case, and this is known as syngamy. Whereas in the second case, the sperm nucleus 2 comes and fuses with the polar nuclei, and this is called triple fusion. So basically 2 fertilization happen, one after another. So now let us understand what happens to the various parts of a flower after double fertilization. So we will start with the parts which directly do not participate in the process of reproduction, as in the non-essential wools. So like the petal, and then the calyx, then the male part and the female part. What happens to them? So the petal, the stamen, which is the androecium, the style the stigma, they all fall off after double fertilization. Next to this is the calyx. Calyx means the sepals. Now these sepals in some fruits and vegetables they may be retained, in some they are lost. Examples of which are, like in case of brinjal they are retained, but in case of apple and guava they dry and they are lost. Then we come to ovary. So this ovary after double fertilization will become the fruit, and the wall of the ovary will become the fruit wall, which is known as pericarp. Next to this is ovule. That means this structure, one structure inside this, the ovule, it becomes the seed. So if a flower has one ovule, there will be one seeded fruit. If it has many, then there will be multiple seeded fruit. Next to this is the placenta. We already know placenta is the structure that connects the ovule with the wall of the ovary. So it will become the stalk of the seed. Next to that, there are two structures, testa and tegument. These are integuments, the outer and the inner integument of the ovule. They form the seed coat. Then comes the secondary nucleus, which secondary nucleus is this structure which is produced right after triple fusion. So this secondary nucleus later on forms the endosperm of the seed. Remember one thing, endosperm, just like cotyledon, is the nutrient rich part of a seed. There is not much difference between endosperm and cotyledon. In some seeds endosperms are retained, in some seeds the endosperms are changed into cotyledons. Then the next part is the egg cell. The egg cell will finally form the embryo, because this is the part where syngamy happens. That means fusion of the sperm nuclei with the egg results into the formation of zygote, and this zygote will divide and redivide and will finally form the embryo. So with this we have finished the chapter pollination and fertilization. If you have any doubts, like I always say, post it in the comment box. Question for you in this video, that what happens to the central cell after double fertilization? If you know the answer, write down it in the comment box. If you have liked my video and if you have liked my teaching, share it with your friends and subscribe to my channel. Thank you.